Imagine that every time you entered one wrong digit in a phone number, your screen cracked. You could keep repairing the screen or correct the digit causing the problem. Now, let's apply this idea to the human body. What if a lifelong disease began with one incorrect letter in our DNA? And what if, instead of taking medicines every day to manage the consequences of that mistake, we could enter the affected cell and correct the letter itself? This is the idea behind Beam Therapeutics, a biotechnology company developing genetic medicines based on a technology called base editing. First, let me remind you of some basics. Inside the nucleus of almost every cell is DNA. The instruction manual used to build and operate the body. Let's say the whole secret recipe. DNA uses 4 chemical building blocks called nucleotide bases. A stands for adenine, T for thymine, C for cytosine, and G for guanine. In DNA, A likes to pair with T, and C likes to pair with G. As the RNA is slightly different, it uses uracil instead of thymine, but let's talk about that later. A section or a part of DNA carrying a biological instruction, let's say a recipe page in a recipe book, is called a gene. When a cell needs that instruction, it makes a temporary messenger RNA copy through a process called transcription. The mRNA leaves the nucleus and travels to a cellular structure called a ribosome. The ribosome reads the mRNA 3 letters at a time. Each 3-letter unit is called a codon. Each codon usually corresponds to a particular amino acid or a stop signal. Amino acids are then connected together like beads on a chain. The amino acid chain then folds into a 3-dimensional structure to form a protein. The second process is called translation. Now, in a simplified form, DNA is transcribed into mRNA and mRNA is translated into protein. It's simple, right? Now, a permanent change in DNA or any DNA sequence is called a mutation. A mutation may replace, insert, or delete nucleotide bases. A substitution involving only one base may have no effect, change one amino acid, creating a missense mutation, or create an early stop signal, creating a nonsense mutation. One altered amino acid can change how a protein folds or functions. The biological chain may then become DNA mutation, abnormal protein, disrupted cell function, and then disease. Now, how does Beam Therapeutics aim to change that? Traditional therapies aim to cut double strands of the DNA where the mutation is happening. It uses CRISPR-Cas9 that comes with a guide RNA and a Cas9 nucleus. A nuclease is an enzyme that cuts DNA. The guide RNA acts like a molecular address. It brings Cas9 to a matching DNA sequence. Once the guide RNA brings Cas9 to the correct location, Cas9 cuts both strands of the DNA double helix, and this creates a double-strand break. The cell must then repair their break. Traditional CRISPR-Cas9 is powerful, but double-strand breaks can sometimes produce unpredictable and unwanted insertions, deletions, or larger DNA rearrangements. Beam uses a next-generation CRISPR-derived technology called base editing. As we said, traditional CRISPR-Cas9 works more like a molecular scissor. Base editing works more like a molecular pencil. Instead of deliberately cutting both DNA strains, a base editor chemically converts one DNA base into another. A base editor contains 3 main functional elements. First, the guide RNA. The guide RNA directs the editor to a specific DNA sequence. Let's say it's like a cab driver. 2, a modified CRISPR protein. Beam uses a CRISPR protein that has been modified so that it does not create a complete double-strand break. Depending on the base editor, this protein may open the DNA and make only a small nick in one strand. It's like a sticker telling the cell, hey, you need to repair this spot and remember it that way. And 3, a deaminase enzyme. The deaminase enzyme performs the chemical modification of the selected DNA base. Let's say it acts like the rubber and the pencil at the same time. An adenine base editor, for example, or ABE, converts an adenine into a molecule called ionosine. The cell reads ionosine as guanine and eventually produces an AT to GC conversion. Concretely, the guide RNA finds the correct address. The modified CRISPR protein opens the DNA, and the deaminase changes the selected letter within a small region called the editing window. The DNA sequence is permanently modified, but the editor itself only needs to be present temporarily. All this to say, traditional CRISPR is like cutting and repairing a sentence. Base editing is more like rewriting selected letters without cutting the sentence in half. Just as we said, correcting the digit that was wrong in a phone number. Now, the gene editing is one part of the challenge. Reaching the correct cell is the second part. Beam uses 2 main delivery strategies. With ex vivo editing, cells are collected from the patient, edited outside the body, tested, and then returned. For blood-forming stem cells, editor mRNA and guide RNA enter the cells through electroporation. It's brief electrical pulses that temporarily open the cell membrane, allowing editor mRNA and guide RNA to enter the cell. With in vivo editing, the treatment is delivered directly into the body. For liver diseases, Beam packages editor mRNA and guide RNA inside lipid nanoparticles. These particles are taken up mainly by hepatocytes, the principal functional cells of the liver. The editor is present only temporarily, but the DNA modification may remain inside the edited cell. Now, where does Beam stand today? Beam is a clinical stage biotechnology company, but it is not yet a commercial pharmaceutical company. Its 2 most highlighted programs are Risto-cel, its most advanced ex vivo program developed for sickle cell disease, and Beam 302, developed for alpha-1 antitrypsin deficiency and its leading in vivo program. Beam is also developing Beam 301 for glycogen storage disease and Beam 304 for phenylketonuria. As of July 2026, no Beam medicine has received commercial approval yet. Beam's central question is simple. Can we identify the responsible cell, gene mutation, and protein, and then safely rewrite DNA to correct the disrupted biological pathway? And that is Beam's promise, treating genetic disease not only by managing its consequences, but by changing the biological instructions that started the problem.